On the screen, a single wave is rising. It moves slowly, far more slowly than the eye expects from any electrical signal, from any living thing. From its leading edge lifts from the flat baseline of the recording and climbs in a long, unhurried curve, the way a deep ocean swell lifts the surface of the water far out to sea where no wind is blowing. It rises for nearly half a second. It keeps rising. By the time it reaches its peak, it stands more than 3 times as tall as any other feature on the trace, 75 microvolts from trough to crest, a measurement that carries real weight here, in a signal where most events are small and brief. Then it falls. It falls for another half second, slowly, with the same unhurried gravity, all the way down through the baseline and below it into a long trough, before climbing again toward the level ground of silence. This is a delta wave. It belongs to a sleeping brain. Everything around it on the recording looks smaller by comparison. The quick flickering of faster rhythms, the occasional brief bursts of faster oscillation nested in its rising edge, the smaller signals of muscle and breath and heartbeat arriving from other electrodes. All of it is dwarfed by this one slow, enormous wave. The brain that produced it is not dreaming. It is not processing language or solving any problem. It is doing something older and quieter than any of that, something the brain has done every night for as long as there have been brains to do it. It has descended to its deepest resting level, and it is moving in waves. The stage this wave belongs to has a name, NREM, Stage 3, or Slow Wave Sleep, and it sits at the very bottom of the architecture the sleeping brain constructs each night. Sleep does not arrive all at once. It comes in layers, in a structure that repeats across the hours of the night in roughly 90-minute cycles, each cycle moving through lighter stages, descending to this deepest one, and then rising back toward the surface before beginning again. In the first 2 cycles of the night, the descent goes all the way down. The brain reaches stage 3. It remains there for stretches of 20 minutes or more. The great slow waves roll across the recording in long, steady processions, one after another, each one as large as the last. The organization of sleep into its distinct stages was formalized in 2007, when the American Academy of Sleep Medicine published updated criteria that unified all deep NREM sleep into a single classification. The threshold is precise. A 30-second window of recording crosses into stage 3, when 20% or more of that window is occupied by slow wave activity, by waves in the range of half a hertz to 4 hertz in frequency, from each exceeding 75 microvolts in amplitude as measured at the scalp above the frontal regions of the brain. Below that threshold, the sleeper is in lighter territory. Above it, they have arrived somewhere else entirely. The criteria give a number to something that was already deeply real, a line between the surface of sleep and its floor. The electrodes that measure these waves sit against the scalp above the forehead, over the frontal lobes, because this is where slow wave activity is largest, where the amplitude is greatest, and the signal clearest. The frontal cortex does not generate these rhythms alone, but it expresses them most powerfully, and it is the first region of the brain to reach the deepest levels of slow wave activity each night. From there, across the wide sheet of the cortex, the waves propagate. Each slow wave is not merely a large electrical fluctuation. It is the surface expression of something happening across the brain simultaneously, something involving hundreds of millions of neurons moving in a kind of shared rhythm. The scientific name for the slow rhythm that underlies the deep sleep EEG is the cortical slow oscillation. Its frequency is lower even than the delta band, around half a hertz to 1 hertz, meaning it completes between 1/2 and 1 full cycle per second, which is to say, 1 full cycle in roughly 1 to 2 seconds. Within each oscillation, the entire population of neurons in a given cortical region passes through two opposite states. First, a depolarizing up state, a period when neurons are active, when the cortex is briefly alive with firing. Then, a hyperpolarizing down state, a period of near complete neuronal silence, when the electrical activity of the cortex collapses and the brain generates, at the scalp, a large slow deflection in the negative direction. These two states together produce what the recording shows, not just a wave, but a full oscillation, a breathing in and a breathing out of neural activity, slow enough to observe with the naked eye on the trace, large enough to dominate every other feature of the signal. The men who first described these oscillations in precise cellular detail were working in the 1990s and early 2000s. Mircea Steriadi and his colleagues spent years recording from neurons deep in the cortex and thalamus of sleeping animals, watching the population alternation between up and down states unfold at the single cell level. Marcello Massimini and colleagues later extended this work to the propagating nature of slow oscillations in the human sleeping brain, revealing that each slow wave is not merely a simultaneous event across all of the cortex at once, but a traveling phenomenon, a wave in the truest physical sense, moving through the cortical surface the way a ripple moves across water. It begins most often in the frontal regions, and it sweeps across the brain from front to back, each region entering its up state and then its down state in sequence, the whole passage taking roughly half a second to cross the full extent of the sleeping cortex. There is something deeply ordered in that image. The sleeping brain, apparently inert, is in fact moving, not in the direction of any task or purpose, but through its own interior, cycling between activity and silence, between firing and stillness, with a regularity that borders on the tidal. Inside the great slow waves, another rhythm is nested. If the EEG trace is inspected closely during stage 3, at the rising slope of a slow oscillation's upstate, a brief and different pattern sometimes appears. A small, fast cluster of oscillations lasting less than 3 seconds, spindle-shaped in appearance, rising from a central peak and tapering at both ends. These are sleep spindles. They are generated not in the cortex but in the thalamus, a paired structure deep in the center of the brain that acts in waking as a relay station for sensory information. During sleep, the thalamus withdraws from that relaying role and begins generating its own internal rhythms, spindles in the 12 to 15 hertz range, brief and regular, erupting in bursts of half a second to 3 seconds, and then falling silent again before the next one forms. The timing of these spindles is not random. They arrive preferentially during the tracking app state of the cortical slow oscillation, during the ascending phase of the large slow wave, when the cortex is momentarily active. This coupling between the thalamic spindle and the cortical slow oscillation is one of the most important features of stage 3 sleep, and its regularity suggests that the two rhythms are not independent. They are coordinated. The slow oscillation, sweeping from front to back across the cortex, seems to open a narrow window of excitability each time it rises, and within that window, the thalamus delivers its spindle. The precise alignment of these two rhythms, and a third shorter rhythm arising from the hippocampus, is thought to be central to what deep sleep does for memory. But the spindles themselves, even before their function is considered, are visible here in the recording as small and transient events, brief tremors in otherwise quiet ground, appearing and disappearing in under 3 seconds, nested in the long rise of the wave. To hold the whole night's architecture in a single image, sleep researchers use a graph called a hypnogram. It is a simple plot, clock time along the horizontal axis, sleep stage along the vertical, with wakefulness at the top, light NREM below it, deeper NREM below that, and REM sleep in its own position above stage 3. When the graph is drawn for a full night of sleep, from the time a person closes their eyes to the time they open them in the morning, it produces a characteristic shape. In the first hour and a half, the trace descends steeply from the surface, from wakefulness through stage 1, through stage 2, and then all the way down to stage 3. The graph drops to its lowest point, and there it stays for a stretch of 20 minutes, sometimes longer, before climbing again into lighter sleep and eventually into a first brief episode of REM. Then it descends again. Roughly 3 hours into the night, after that first REM period, the trace drops down once more into stage 3, though not quite as deeply, not quite as long, the delta waves perhaps slightly reduced in their amplitude, the N3 epoch briefer than the one before. By the middle of the night, the balance has shifted. Stage 3 appears less. REM periods grow longer. The graph no longer drops all the way to its lowest point in the later cycles of the night. It hovers in the middle range, spending more time in lighter NREM and more time in REM, the first great N3 valley of the early night, now the deepest the graph will reach. There is a remarkable economy in this distribution. The brain concentrates its deepest work in the first half of the night, front-loading the slowest, largest amplitude waves into the hours immediately after sleep begins, and then easing gradually upward as the night progresses. By the time the first light of morning appears beyond a window, the hypnogram's last full cycle has brought the trace only to stage 2, and REM, the deep valleys of the early hours long past. The brain has already done most of what slow-wave sleep does. The deepest architecture of the night exists in those first 2 descents. The 30-second epoch, the scoring unit, the basic tile from which the hypnogram is assembled, is the fundamental measure of this territory. A polysomnographic recording of a full night of sleep might contain nearly 1,000 such epochs. Each one is examined for the presence of slow-wave activity. Each one that crosses the 20% threshold is classified as stage 3. Laid in sequence, they form the valleys in the hypnogram, the deep early night descents that mark the times when the sleeping brain was moving at its slowest and most powerful rhythm. The amplitude of those waves at the frontal electrodes is not fixed. It varies with how long the person has been awake before sleep begins. Sleep pressure, the accumulated drive for sleep that builds during waking hours, has a direct and measurable effect on slow-wave amplitude. After a long day, or after a night without sleep, the first stage 3 epoch of the following night contains delta waves that are taller, more regular, and more densely packed than those of a baseline night. The brain arrives at deep sleep, carrying the weight of its prior wakefulness, and releases it in the form of larger, slower, more powerful waves. As the night continues and slow-wave sleep accumulates, that pressure is discharged, and the waves in the later epochs are smaller, the stage 3 epochs shorter, the descent less steep. The hypnogram's valleys are not merely a clock. They are a register of what the brain was owed and what it has repaid. The brain has been producing these rhythms for a very long time. Sleep in some form appears in almost every animal that has a nervous system complex enough to sustain it. The slow oscillations of mammalian NREM sleep, the alternation between up states and down states, The large delta waves in the cortical recording are not uniquely human. They have been measured in cats and rats and ferrets, in recordings taken in the 1960s and the 1990s, and the present decade. The fundamental architecture appears to have been conserved across the long history of mammalian life, a feature of the sleeping brain so deeply embedded that it persists across hundreds of millions of years of divergent evolution. The specific circuits may vary. The duration of the sleep period may differ enormously between a mouse and an elephant, but the slow wave, the great oscillation between upstate and downstate, the large slow deflection at the scalp, appears wherever there is a cortex and a thalamus and a night. This does not mean it is simple. The machinery that generates a delta wave involves some of the most complex cellular interactions in the nervous system. Thalamic neurons switching modes, cortical populations coordinating across wide distances, inhibitory interneurons shaping the timing of every cycle. But that is the machinery beneath the signal. The signal itself, as it appears on the recording trace, is among the simplest things the brain produces. A long, slow, large wave, rising and falling at less than 4 cycles per second, its amplitude dwarfing everything around it, its rhythm as steady and slow as breathing. The first of those waves appears very quickly after a person falls asleep. The transition from wakefulness to stage 1 to stage 2 can take only a few minutes in someone who is sufficiently tired. Stage 2 brings its own rhythms, spindles and K complexes, the hallmarks of medium-depth sleep. And then, somewhere between 15 and 30 minutes after sleep begins on most nights, the descent continues. The Ka complexes grow more frequent. The spindles begin to appear against a background of increasing slow-wave activity. The amplitude of the EEG climbs, the frequency drops, and then the 20% threshold is crossed. The epoch that contains that crossing is scored as stage 3. The person lying in the dark with their eyes closed has arrived at the bottom of the architecture their brain constructs each night, and the great waves have begun. For the next 20 minutes or 30 minutes, or sometimes more, on this first descent of the night, the recording is dominated by those waves. They are not perfectly regular. Some are larger, some slightly smaller, some arrive in close succession, some with a brief pause between them. But the general character of the trace is unmistakable. Slow, high amplitude, front-weighted, rolling. The brain that produces them is not processing the outside world. It is not laying down explicit memories of what is happening in the room. It is sealed from the environment in a way that no other stage of sleep quite matches. Deeper even than REM, more profoundly disconnected from incoming information. A sound that would reliably wake this person from stage 2 will be absorbed without response in stage 3. A voice calling their name from the doorway may produce no reaction at all. The brain continues generating its slow waves, and the waves say nothing about the sound, and the person does not wake. What the recording cannot show, and what no single trace can fully convey, is the scale of what is moving through the brain when those waves appear. The amplitude at the scalp is the summation of the electrical activity of an enormous number of neurons, millions of them, spread across the cortical surface, all entering their upstates at nearly the same moment, all falling into their downstates in the same brief interval. The size of the wave reflects the size of the coordination. A larger wave means a larger population of neurons moving together. The frontal cortex generates the largest waves, not because it is the only region participating, but because its neurons coordinate most completely, their rhythms most tightly aligned. At the back of the brain, the occipital cortex, the region that processes vision, generates smaller, slow waves, its population less synchronous, its contribution to the overall rhythm less dominant. But the wave does reach it. The traveling wave that begins at the frontal cortex sweeps backward across the brain, arriving at each region in sequence, carrying the slow oscillation with it. By the time it reaches the back of the head, the frontal cortex has already begun its next up state. The brain is not firing all at once in deep sleep. It is cycling through a repeating wave, a traveling rhythm that crosses the cortical surface from front to back, again and again, at roughly once every second or two, for as long as stage 3 continues. The image this creates, if it were possible to watch the sleeping brain's electrical activity from above, to see all of the cortex simultaneously, each region colored by its current state, would look like a slow tide moving across a shallow sea, a brightening at the front, spreading backward, dying away at the rear, then another brightening beginning at the front again, the wave crossing the full width of the brain before the next one starts. Slow, regular, vast in its coordination, indifferent to everything happening on the other side of the skull. The polysomnographic record, the full recording of brain waves, eye movements, muscle activity, and breathing that sleep researchers use to study these stages, captures this in a compressed form. Across 30 seconds of paper or screen, the deep sleep record shows the large biphasic deflections of the slow oscillations, the occasional nested spindle, visible as a brief high-frequency burst against the low-frequency background, the regular rhythm of breathing expressed as slow undulations in the respiratory channel, the absence of eye movements that characterizes all NREM sleep. Everything in the record is quiet except the brainwaves themselves, and those are enormous. The scoring of that record, the assignment of each 30-second epoch to a sleep stage, is the work of a trained scorer following the 2007 criteria. In practice, stage 3 epochs are not difficult to identify. The delta waves are large enough that they are visible at a glance. The question is not whether they are present, but whether they occupy enough of the epoch, whether 20% or more of those 30 seconds is given over to slow wave activity. In deep early night sleep, that threshold is usually far exceeded. The delta waves fill the epoch almost completely, sometimes accounting for 60 or 70% of the signal, leaving only brief interstices of faster activity between them. Late in the night, in the third and fourth sleep cycles, stage the third of may not appear at all. The hypnogram stays in stage 2, and REM, the deep valleys of the early hours, unrepeated. The brain has discharged most of its slow wave sleep pressure in the first half of the night, and what remains is the lighter architecture of the later cycles, the longer REM periods, the briefer stage 2 stretches, the gradual approach of morning. It is in this way that the night's architecture has a shape that is not arbitrary. The distribution of sleep stages across the 8 hours of a full night is not random. It follows a pattern that appears to be regulated, that changes predictably with sleep pressure and prior sleep history, that varies in consistent ways with age and illness and pharmacology. The two deep valleys of the early night, the earliest and deepest descents of the hypnogram, are the brain's primary investment in slow-wave sleep. Everything that happens in stage 3, everything the slow waves accomplish in the brain and body, is accomplished mostly in those first 2 descents, in the first 3 to 4 hours of the night. By the time the final cycle of sleep has begun, long before the alarm sounds or the light grows, the brain has already traveled this territory. The delta waves of the first descent, those great slow waves rising to 75 microvolts above the frontal cortex, rolling across the brain from front to back, cycling between upstate and downstate at less than 1 hertz, are long past. They have come and done whatever deep sleep does, and the brain has moved on, cycling upward through lighter stages, spending more time in the dreaming state of REM, preparing in its own quiet way for the approach of waking. The hypnogram records this entire movement. Plotted against the clock, it shows the night as a series of descents and ascents, the steep early drops, the gradual shallowing across the middle hours, the final cycles hovering near the surface. The two deep valleys of stage 3 stand out from the rest of the graph, the way the deepest trenches on the seafloor stand out from the surrounding ocean bottom. Not merely deeper, but categorically different, a different kind of territory from the lighter regions above them. Those valleys are shallower on the second descent than on the first. The first N3 epoch of the night is typically the longest and most intense, the delta waves at their tallest, the stage persisting for the greatest duration. By the second descent, the waves are still large enough to cross the scoring threshold, the slow wave activity still dominant across the epoch, but the amplitude is beginning to ease. The brain's most urgent slow wave work has already begun in the first descent, and the second is a continuation rather than a repetition. Across the third and fourth cycles, the N3 valleys fade from the graph entirely, replaced by the flat line of stage 2 and the distinctive shallow arch of REM. By the fifth or sixth cycle, in those final hours before morning, the hypnogram moves only between stage 2 and REM and brief awakenings. Stage 3 is gone. Its time is past. The great waves are no longer moving across the cortex, and yet the night is not yet over. The sleeping brain continues its work in lighter territory. The shorter cycles of REM, the brief stage 2 interludes, the system still operating in the organized darkness, while the body rests against the warmth of a mattress, breath slow and regular, the room unchanged since the night began. The two valleys, those deep early night descents, the first one deepest, the second slightly shallower, represent the full extent of stage 3's residence in a healthy night of sleep. They are brief in the context of 8 hours. They are concentrated in a specific window. They are precisely timed and precisely measured, and they are, by the criteria of the sleeping brain's own electrical record, the deepest, slowest, most organized states the brain ever enters. On the hypnogram, they appear as a simple shape, a steep descent, a brief floor, a return. But behind that shape lies an architecture of enormous complexity and precision, a system that has been refined across the entire history of mammalian life, that deploys the largest electrical events the brain produces, that coordinates the activity of tens of millions of neurons across the cortical surface, with a timing accurate to the millisecond. All of this becomes, on the graph, two quiet valleys in the early night, growing a little shallower as morning draws closer, the brain completing its deepest work before the light returns. There is a kind of quiet wonder in that economy. The brain does not distribute its slow-wave sleep evenly across the night, spreading it thin across all 8 hours, making it available at any time. It concentrates it, it front-loads it. It delivers the deepest and most powerful stages first, when the pressure for them is greatest, and spends the rest of the night in lighter territory. The hypnogram tells this story in a single glance, the architecture of a sleeping brain, all of it visible in the shape of a line, the two deepest valleys of the early night, the most important part of the record, and already behind the sleeper, long before the alarm sounds. Beyond the window, nothing has changed since the night began. The room holds its stillness. The air is the same temperature it was an hour ago. The darkness is complete, and the brain that produced those great slow waves in the first hours of the night is still running, lighter now, closer to the surface, but still organized, still cycling, still doing what the sleeping brain has always done through all the long dark hours before morning. The first deep valley has already passed, the second has come and gone, and the slow architecture of the night moves on, shallowing toward dawn, the deepest work of sleep quietly complete. Deep inside the thalamus, where the brain's sensory relay sits in a small paired mass no larger than a walnut, something is changing. For hours of waking life, each half of this structure has worked faithfully, receiving the world's signals from the eyes and the skin and the ears, sorting them, passing them upward to the cortex in continuous streams. Sound and light and pressure have moved through it like traffic through a junction. Now the traffic is stopping, not because the signals have ceased, but because the gate itself is closing. The relay neurons inside the thalamus go quiet in a particular way. Their membranes, which during waking maintained a steady voltage close to a threshold of firing, begin to hyperpolarize, the electrical charge across each membrane shifting further from the level needed to fire. This shift does not come from outside the cell. It comes from channels embedded in the membrane itself, channels that respond to the very voltage they help to create, opening and closing in slow cycles driven by the cell's own chemistry. Two conductances interact here in the dark. One is a slow inward current carried by sodium and potassium ions that activates when the membrane is hyperpolarized, a current that begins to depolarize the cell again from within, not waiting for anything from the world outside, but generating its own slow drive toward the threshold. The other is a calcium current, low threshold, activating only when the membrane is already partially hyperpolarized, producing a small inward surge of calcium ions that carries the cell the rest of the way to firing. Together, these two conductances create a rhythm. The cell hyperpolarizes, and the first current turns on. It nudges the membrane upward toward threshold. The calcium current catches the movement and amplifies it. The cell fires in a brief burst. Then it hyperpolarizes again, and the cycle continues, slow and internal, driven from within. This is burst mode. The relay neuron is no longer a faithful messenger of the outside world. It has become a slow internal oscillator, firing in the rhythm of its own making, rather than in response to anything arriving from the senses. The world's signals still arrive at its dendrites. They find, for the most part, a cell too deeply hyperpolarized to respond. Around the thalamic relay cells, like a shell, lies a structure of inhibitory neurons that wraps most of the thalamus in a thin curved sheet. Every relay cell that sends its axon upward to the cortex sends a branch back through the shell, activating its inhibitory neurons as it goes, and those inhibitory neurons project back onto the relay cells themselves. During waking hours, this feedback loop is kept under control. Wake-promoting neuromodulators, released from distant brainstem nuclei, arriving like a standing tide, keep the inhibitory shell neurons from dominating. Norepinephrine, acetylcholine, histamine, these chemical signals, each arriving from its own source, each maintaining the cortex and thalamus in a state of broad excitability, decline as slow-wave sleep deepens. They do not simply become less active. They withdraw, one system at a time, in a sequence that is itself one of the signatures of falling into the deepest sleep. As each withdraws, the inhibitory shell is released. Its neurons fire more freely. Their GABA B receptor-mediated inhibitory signals, slow, sustained, powerful, wash over the relay cells below them, extending and deepening the hyperpolarization. Burst mode becomes more stable. The gate through which the world entered stays more firmly closed. A sound arrives at the sleeping ear. The cochlea translates it into electrical signals. The auditory nerve carries those signals toward the brainstem, and they travel onward toward the thalamus. They reach the relay neurons in the auditory thalamus and find membranes in deep hyperpolarization, inhibitory currents sustaining their silence. The signals are present but cannot pass. The cortex does not hear the sound. The gate was shut before the sound arrived and remains shut after it is gone. What the cortex generates instead is something of its own. In the neocortex, the same withdrawal of waking neuromodulators that freed the thalamic inhibitory shell has left the cortical membrane in a state of quiet. Pyramidal neurons, the long-range projection cells that make up the cortex's main output, sit at a resting potential a little below the threshold needed for firing. Their excitatory connections to each other, normally humming with activity during wakefulness, have grown quiet. And out of this quiet, something begins. A group of cells in the frontal cortex, for reasons that are still not entirely clear, depolarizes. The membrane voltage rises across a population of neurons, moving toward threshold in a broad coordinated shift. The cells begin to fire. Their axons carry excitation to neighboring cells, and those cells begin to fire as well. Excitation spreads through local connections, jumping from neuron to neuron across the surface of the cortex, each active cell recruiting more. This is the upstate, a period lasting roughly half a second, during which neurons across a wide region of cortex fire at rates not unlike the rates they reach during active waking. In an EEG recording made from electrodes on the scalp above, this moment of collective firing appears as a large upward deflection, the voltage rising as the combined charge of thousands of active cells lifts the recording above baseline. Within the upstate, nested fast oscillations appear and disappear. Sleep spindles, bursts of 12 to 15 cycles per second, generated within the thalamus by a separate mechanism and sent up to the cortex, arrive during this period of heightened excitability, riding the rising slope of the slow oscillation like a small tremor passing over a long, slow wave. A spindle lasts half a second, or 1 second, or sometimes 2, a brief flurry of faster rhythm inside the dominant slow architecture of the epoch, appearing in the EEG as a brief spray of higher frequency oscillation superimposed on the broad curve of the slow wave. Then it is gone. The upstate continues for its remaining duration, the pyramidal neurons still firing, the excitatory current still passing through local circuits, and then the upstate ends. This ending is not external. Nothing turns it off from outside. The very activity of firing during the upstate generates the conditions that terminate it. Across the firing population, excitatory synapses that have been releasing their transmitter continuously begin to exhaust their immediately available vesicle pools. Calcium has been entering neurons and accumulating in cytoplasm and mitochondria, activating calcium-dependent potassium channels whose outward current opposes depolarization. Inhibitory interneurons, small, fast parvalbumin-positive neurons that monitor the activity of the pyramidal cells around them, have been recruited throughout the upstate and now fire strongly, providing powerful inhibitory input around the cell bodies of the very neurons they surround. Their GABA-mediated inhibitory currents arrive at the pyramidal cell membranes and push the voltage downward. The excitatory drive is failing from within. The inhibitory pressure is rising from the interneurons around each cell. Both forces move in the same direction, and within a few hundred milliseconds of each other, the neural population collapses. This is the down state. It is not passive quiet. It is an active hyperpolarization, a state during which the membrane voltage across the cortical population is held 10 or more millivolts below the resting potential, a gulf that leaves the cells genuinely unable to fire, even if excitatory signals arrive. GABA B receptors, slower and longer lasting in their inhibitory effect than their GABA A counterparts, are activated by the interneuron's output and sustain this hyperpolarization for several hundred milliseconds, the inhibitory current outlasting the inhibitory firing that produced it. The cortex is silent, truly actively silent, not simply unoccupied, but held below threshold by chemical forces running their course. In the EEG record, the downstate appears as a large downward deflection, the population voltage dropping well below baseline, the trace swinging low. This is the other half of the slow oscillation shape, the down deflection following the up deflection, the trough following the crest, together forming the large biphasic wave that is the defining mark of deep sleep on an EEG trace. From crest to trough, and back to the beginning of the next crest, the full cycle takes roughly 1 second at its most characteristic frequency, a rhythm cycling at about once per second, sometimes a little slower, sometimes a little faster, filling the sleep epoch with great slow undulations that move the EEG trace up and down far more than any other electrical event the brain produces during the night. Across the sleeping brain, this cycle does not happen all at once in every region simultaneously. The slow oscillation travels beginning in the frontal cortex, where it is most reliably generated and most densely expressed. The upstate propagates as a wave across the cortical surface, sweeping toward the posterior cortex over the course of roughly half a second. One region depolarizes into its upstate and recruits the next, and the next recruits the region beyond it, so that the wavefront of activation moves in a roughly front-to-back direction across the brain, reaching the occipital regions after it has already peaked and begun to fall away in the frontal areas where it started. This directional propagation is consistent and reliable enough that it can be mapped across the cortical surface of a sleeping person using a dense array of scalp electrodes, the slow wave visible as a traveling pattern rather than a stationary event. The thalamus participates in this traveling wave. As the cortical upstate sweeps through a region, the excitatory connections from cortex to thalamus carry some of that activity downward, briefly depolarizing the thalamic relay neurons in that region. They join the upstate for a moment. They may fire a burst. Then, as the cortical downstate arrives, the thalamic cells are pulled back into hyperpolarization, and the inhibitory shell is reactivated, and the relay neurons return to burst mode, their internal chemistry once more generating the slow rhythmic oscillation that will sustain the next sleep cycle. This coordination between cortex and thalamus is sustained without conscious control, without any external regulator. The two structures communicate continuously through reciprocal connections, cortex exciting thalamus, thalamus projecting back to cortex, and their interaction during deep sleep produces the synchronized large amplitude oscillations that define the EEG record of this stage. Neither structure alone could maintain what they produce together. The cortex generates the slow oscillation from its intrinsic cellular properties. The thalamus amplifies it through its own burst mode firing. The inhibitory shell sharpens and times the transitions. Together, they sustain a rhythm so large, so slow, and so precisely coordinated that it appears in the EEG above a sleeping skull as waves easily visible to the naked eye. Great slow swells crossing a centimeter per second across the recording screen. From a frontal scalp electrode, an FFT spectrum computed across a 30-second epoch of stage 3 sleep shows a single sharp peak towering above all other frequencies. It rises steeply from the baseline at frequencies below half a cycle per second, reaches its apex somewhere between 1 and 3 cycles per second, and falls away as the frequency increases beyond 4 cycles per second. This peak represents the delta band, the mathematical fingerprint of slow wave activity extracted from the EEG record through Fourier analysis, which decomposes the complex waveform of the EEG into its constituent frequencies and measures the power in each. The area under the curve within the delta band is what researchers call delta power. It is the primary quantitative measure of homeostatic sleep pressure, the accumulated biological need for sleep that builds across every waking hour and discharges across each period of slow wave sleep. The relationship between prior wakefulness and delta power is direct and consistent. The longer the preceding waking period, the greater the delta power measured in the first slow wave sleep episode of the subsequent night. This is not a rough correlation, but a proportional relationship, precise enough to be formalized in a mathematical model. Alexander Borbély, working through the 1980s, proposed a framework for understanding sleep regulation in which two independent processes interact. The first is a circadian signal, the 24-hour biological clock in the hypothalamus, which promotes wakefulness during the day and sleep during the night, regardless of how much sleep has been obtained. The second is a homeostatic process, a drive for sleep that accumulates during waking and dissipates during sleep, tracked by the brain with what appears to be considerable fidelity. Delta power is the best available readout of this homeostatic process. When it is high, the brain's accounting of its own sleep debt is visible in the EEG, written in the amplitude of the slow waves rolling through the frontal cortex. When it falls across the night, the account is being settled, the biological pressure for deep sleep gradually discharged by the very oscillations it demands. This relationship between prior waking and the depth of subsequent slow wave sleep, between neural firing and the inhibition it generates, between the upstate and the downstate it creates, runs through every level of the system. The cortical neuron fires during the upstate, and in firing generates the synaptic fatigue and inhibitory pressure that force it into silence. The inhibitory interneuron is recruited by the pyramidal cell's activity and fires to end the very episode that activated it. The thalamic relay neuron is depolarized into burst mode by the same hyperpolarization that silences it as a sensory relay. At each level, the mechanism that generates the oscillation is the same mechanism that sustains it. The state creates the conditions for its own renewal. The downstate takes hold. Across the frontal cortex, the population voltage lies well below baseline. The inhibitory currents sustaining the hyperpolarization are still running, but beginning to wane. Calcium-dependent potassium channels that helped push the membrane downward are closing as intracellular calcium is slowly removed. The slow inward depolarizing current that responds to hyperpolarization is beginning to activate again, pressing the membrane upward from below. Thalamic relay neurons in their burst mode are generating slow depolarizing waves of their own, sending gentle excitatory pulses up to the cortex as they fire their burst rhythms. And somewhere in the frontal network, the balance tips. A population of cells reaches the threshold again. The upstate begins. The slow oscillation has completed one cycle and started the next. In the EEG, above the sleeping skull, the great slow wave crests again. It will crest roughly once per second for the rest of the first deep sleep episode, and then again during the second episode later in the night. Each wave carries within it the same architecture, the collective depolarization of cortical neurons, the brief spindle nested in the upstate's rising slope, the collapse into population silence, the held hyperpolarization, and the return. Each wave is both the same and slightly different from the one before it, the amplitude varying, the duration shifting within narrow bounds, the propagation path across the cortex not perfectly identical each time. The rhythm is real and powerful without being mechanical, the brain generating its deepest electrical activity through a process that is self-sustaining and self-terminating at every cycle. The parvalbumin positive interneuron fires one more tight burst around the body of a pyramidal cell as the upstate reaches its peak. It is a small neuron, compact and fast, its axon branching close to the cell body of the pyramidal cells it contacts, the most powerful anatomical position for inhibitory control. Its GABA A receptor mediated currents arrive at the pyramidal cell's soma within milliseconds of its firing, shunting depolarizing current before it can accumulate. Its activity during the upstate constrains how long the firing episode lasts and how intense it becomes, imposing a ceiling on the cortical activation that keeps each upstate within the narrow temporal window characteristic of the slow oscillation. Without this constraint, the upstate might expand. The cortex might approach something like a waking firing pattern. The rhythm might destabilize. The interneuron maintains the precision of the cycle. Its brief bursts the inner mechanism by which the brain keeps its own deepest rhythm within bounds. There is a sense in which all of this takes place in something that resembles isolation. The thalamic gate is closed. The cortex is cut off from the sensory world by the inhibitory shell, and the withdrawal of wake-promoting neuromodulators. The great slow oscillations propagate across the cortical surface in the dark, unseen, unheard from outside, generated entirely from within the chemistry of membranes and the physics of ion gradients maintained across the walls of cells. No external signal drives them. No conscious process monitors them. They arise because the conditions for their arising are present: the right cells, the right connectivity, the right ionic conductances, the right balance of excitation and inhibition, and they sustain themselves because each state generates the conditions for the next. This is what the brain at its deepest looks like from the inside, not silent, not still, but rhythmically and precisely active in a way that requires no input and serves no immediate purpose. The cells themselves can know. A sleeping adult's frontal cortex at this moment generates slow waves whose peak-to-peak amplitude routinely exceeds 75 microvolts as measured at the scalp, which means that at the cortical surface itself, the amplitude is substantially larger. The skull and the soft tissue between the cortex and the electrode, attenuating the signal by the time it reaches the recording site. These are among the largest electrical events the sleeping brain produces. They dwarf the low amplitude, high frequency signals of waking. They dwarf the theta rhythms of the transition into sleep. They tower above the background EEG like great swells on an otherwise moderate sea. Their slow frequency and enormous amplitude making them visible at a glance to any experienced sleep researcher scrolling through a polysomnographic record. At the deepest point of each downstate, the cortex produces near-complete neuronal silence across vast regions simultaneously. If a small electrode were placed directly against the cortical surface at this moment, the local field potential would show the large negative voltage deflection corresponding to the membrane hyperpolarization of the surrounding cells, and single-unit recordings of individual neurons would show their firing rates dropping to zero, not nearly zero, but zero, a genuine absence of spike activity sustained for hundreds of milliseconds across a wide neuronal population. This is not sleep as mere inactivity. It is sleep as a profoundly organized biological state, The brain's chemistry, maintaining a precise alternation between collective firing and collective silence, cycling at roughly 1 cycle per second throughout the episode. The system neither drifting randomly nor firing chaotically, but oscillating with the steady, measured rhythm of something built to do exactly what it is doing. Beyond the frontal cortex, across the parietal and temporal regions through which the slow oscillations traveling wavefront passes on its way to the posterior brain, the same upstate and downstate transitions occur with a short delay behind the frontal initiation. The occipital cortex, the most posterior region, receives the wavefront last perhaps 400 or 500 milliseconds after it first appeared at the frontal pole. By the time the downstate is ending in the occipital cortex, the frontal cortex may already be beginning the next upstate. The brain does not oscillate as a single synchronous block. It oscillates as a propagating pattern, front to back, each region cycling with the same intrinsic mechanism, but each passing through the cycle slightly after the region behind it. The traveling wave is real and consistent enough to be measured, its direction and speed across the cortical surface now well established in human sleep recordings. The thalamic relay neurons, their membranes held in hyperpolarization by the sustained inhibitory output of the surrounding inhibitory shell, fire their slow internal burst rhythms in the quiet between the cortical slow oscillations upstates. In the intervals when the cortex is in its downstate, the cortical excitatory drive to the thalamus is absent, and the thalamic cells are left to their own internal pacemaking, their burst mode oscillations continuing at a frequency slightly faster than the cortical slow oscillation, generating a rhythm that will contribute to spindle generation and that will feed back into the cortex as a source of the depolarizing current that helps initiate the next up state. The thalamus and cortex are in constant dialogue, even in the depths of sleep, each providing to the other what the other needs to maintain the cycle. The inhibitory shell, that thin curved sheet of GABAergic neurons wrapped around most of the thalamus, fires in response to the relay neuron's bursts, projecting its inhibitory signals back onto the relay cells and sharpening the transitions between burst and silence. Its neurons are well positioned to synchronize the relay cells within a thalamic region, ensuring that when one relay cell fires its burst, its neighbors receive a brief inhibitory pulse that resets their membranes and brings them into phase. This synchronizing function contributes to the large amplitude, coordinated nature of the thalamic rhythms during deep sleep, the inhibitory shell acting as a pacemaker by enforcing timing through inhibition, a mechanism that coordinates the output of many cells into the coherent rhythm that the cortex then receives. All of this machinery, the thalamic relay cells in burst mode, the inhibitory shell synchronizing their output, the cortical pyramidal neurons cycling between up and down states, the parvalbumin interneurons constraining the up state's duration, the GABA B currents holding the down state in place, the traveling wave sweeping from front to back across the cortical surface. All of it operates in the deep background of a sleeping body. The heart continues its slower nighttime rhythm in the chest below. The breath moves in and out, slow and even. The muscles are relaxed, the posture unchanged, the face still. Nothing in the outward appearance of the sleeping person indicates that a few centimeters inside the skull, a billion neurons are cycling through one of the most precisely organized and powerfully rhythmic electrical states that the brain ever enters. An EEG trace, by contrast, would be unmistakable. The slow waves roll across the recording at nearly 1 per second, each crest and trough so large relative to the signals beside them that they set the scale for everything else. A sleep spindle appears in the slope of one up state, 12 to 15 cycles per second, a brief flurry, then gone, and the slow wave continues as though uninterrupted. The trace is a map of an interior landscape entirely invisible from outside, the electrical record of a brain doing something that takes roughly an hour and a half to arrive at and will sustain itself for 30 or 40 minutes before giving way to lighter sleep, and eventually to a brief period of REM before the cycle begins again. The slow oscillation does not stop between cycles. As the first deep sleep episode ends, the oscillation gradually loses amplitude. The delta waves become smaller, less regular, less frequent. The proportion of the epoch given over to slow wave activity drops below the scoring threshold. The sleeper ascends toward lighter sleep on a schedule that is itself homeostatic. The first deep sleep episode, having discharged the most acute component of the accumulated sleep pressure, the delta power measured at its end somewhat lower than at its beginning. The second episode, 90 minutes later, will begin with less initial delta power than the first, its slow waves still real, but lower in amplitude and shorter in total duration. The homeostatic discharge continues across the night, each successive episode of deep sleep, drawing down the reservoir a little further until by the early morning hours, the deep sleep episodes are brief or absent, and the brain fills its remaining cycles with lighter NREM sleep and longer stretches of NREM. The relationship between delta power and homeostatic pressure has been studied extensively in both normal sleep and in sleep deprivation, and it holds with remarkable consistency. Total sleep deprivation for a night produces a first recovery episode with dramatically elevated delta power. The waves higher, the density greater, the slow wave proportion of the epoch well above typical baseline values. Selective suppression of slow wave sleep, achieved in experimental settings by playing tones calibrated to disrupt deep sleep without waking the sleeper, produces a compensatory increase in slow wave activity in the subsequent recovery night, suggesting that what the homeostatic system tracks is not simply total sleep time, but specifically the amount and intensity of slow wave activity. The brain keeps an account of its own deepest sleep, and it keeps that account accurately. There is something particular about the precision of this accounting. The faithfulness with which the delta power in the first sleep episode reflects the duration of the preceding wakefulness, the consistency with which the slow oscillation propagates from front to back across the cortex, the reliability with which the pervalbumin interneuron constrains the upstate within its characteristic window. The machinery of deep sleep is not approximate. It is calibrated to a degree that only becomes visible when it is perturbed. When the waking period is extended, or the delta waves are suppressed, or the thalamus is selectively targeted, and the system responds with a restoration of exactly what was missing, no more and no less. The brain does not overshoot. It does not undershoot. It settles the account. In the dark of the sleeping skull, the traveling wave of the slow oscillation is beginning another sweep. It appears first in the frontal cortex, as it always does, the population voltage rising as a cluster of neurons tips into their upstate and recruits the network around them. It spreads backward across the brain in its characteristic half-second traverse, each region lighting briefly into coordinated firing before the downstate follows and the silence is held. The inhibitory currents are running their course. The membranes are preparing, almost imperceptibly, for the return. Somewhere deep in the thalamus, a relay neuron whose membrane was hyperpolarized into its own slow internal rhythm fires a brief burst, sending a small pulse of excitation upward along its axon toward the cortex above. A quiet signal, barely enough to shift the balance, but arriving at exactly the moment when the cortical downstate is beginning to release its hold and the next upstate, the next slow wave, is already beginning to form. The wave crests, the trace rises, and the great slow rhythm of the sleeping brain continues in the dark, cycling in its own precise and self-sustaining order. As the night moves forward and the body lies still, cerebrospinal fluid moves through the sleeping brain along channels that run beside the blood vessels, through narrow corridors of living tissue, in a slow and steady current that no one knew existed until only recently. The brain's interstitial space, the fine fluid-filled gaps between cells, expands by more than half during sleep, opening the way for this fluid to move with a speed and purpose impossible during waking hours. What it carries away is the residue of thought itself, the molecular debris left behind by a day of firing neurons, the protein fragments that accumulate in the spaces between cells, the way sediment settles to a riverbed. Among those fragments are two proteins, amyloid beta and tau, whose buildup across years and decades is linked to the slow erosion of memory and function that defines some of the brain's most serious diseases. During deep sleep, they are carried away in the current. The brain, in its most silent hours, is cleaning itself. The channels through which this fluid flows run along the walls of blood vessels, guided by long, fine processes extending from the brain's supporting cells, the astrocytes, whose feet press flat against the vessel walls and form a lining through which the fluid is drawn. In the sleeping brain, these channels open wider. The cells themselves appear to shrink slightly, pulling back from one another, and in the space they leave, the fluid moves faster, penetrating deeper into the tissue it serves. The mechanism is still being mapped, but the result is measurable. The clearance of waste during sleep is dramatically faster than during waking, and the difference is not small. The current runs at a pace that cannot be matched while the brain is occupied with the world. There is something calm in the image of it, a river running through the dark of the skull, carrying nothing that is needed, only what must be let go. The fluid moves in silence. No signal initiates it. No conscious process oversees it. Deep within the tissue of a sleeping brain, the work of clearance proceeds the way a tide moves through a salt marsh, not because anything decides it should, but because the conditions for it have arrived. Those conditions are created by slow-wave sleep itself. The compressed, metabolically intense hours of waking activity leave their mark on the brain's internal environment, in accumulated proteins, in oxidized molecules, in the concentration gradients that shift across cell membranes with every burst of electrical activity. The slow oscillations of deep NREM sleep, in their large rhythmic sweeps, appear to drive the fluid exchange that clears this mark away. Each slow wave is a broad electrical event that passes through millions of neurons at once, and its passage through the tissue may help pulse the interstitial fluid forward, the way a gentle squeeze moves fluid through a sponge. The mechanism is not fully settled, but the relationship is clear. The slowest, deepest electrical activity of the night corresponds to the fastest movement of the cleaning current. The brain's most restful state is also its most diligent. Somewhere in the first hour of sleep, still early in the night's deepest descent, a small gland at the base of the brain releases the largest pulse of growth hormone it will produce across the entire day. The pituitary gland, barely the size of a grape and tucked into a bony recess at the skull's floor, sends a single biochemical wave outward into the bloodstream, a signal that travels in all directions at once, reaching muscles and bones and organs within minutes. What it says, in the language of molecular biology, is that the time for repair has come. Protein synthesis can begin. Damaged tissues can be rebuilt. The metabolic business of growth, not only in the young, but in the ongoing maintenance of every living body, can now proceed in full. The connection between this hormone pulse and slow wave sleep is precise enough that when the first deep sleep episode is experimentally suppressed, the hormone pulse is delayed or reduced in proportion. Deep sleep is not merely a permissive condition for growth hormone release, it is what triggers it. The first episode of NREM Stage 3 is the key that opens the repair cycle, and the body has been waiting through every waking hour for that key to turn. Growth hormone's reach extends far. It travels to skeletal muscle, where it promotes the uptake of amino acids and the synthesis of new contractile proteins from the day's exercise. It signals to bone, where it stimulates the cells responsible for mineral deposition and the slow continuous remodeling that keeps bone dense and resilient across a lifetime. It acts on the liver, directing metabolic resources away from the immediate demands of waking activity and toward the longer horizon work of structural maintenance. And it regulates fat metabolism in a way that shifts energy balance across the recovery period. The body burning stored fat while building protein, the composition of tissues changing quietly while the conscious mind has no awareness that anything is happening at all. The pulse lasts only a short time, then it passes and the gland goes quiet. But what it has set in motion continues across the remaining hours of the night. Within the neurons of the cortex during deep sleep, a different kind of restoration is underway, one that operates not at the scale of organs or tissue, but at the scale of molecules inside single cells. The waking brain is an energetically expensive place. A firing neuron burns oxygen and glucose at a rate that cannot be sustained without interruption. Across the waking day, byproducts accumulate inside cells, oxidized proteins that have been damaged by the reactive molecules produced during energy metabolism, worn mitochondria that have degraded from their peak efficiency, debris from molecular processes that turn over constantly and leave fragments behind. These are handled slowly, imperfectly during waking hours, but the demand outpaces the capacity for maintenance while the brain is occupied with being conscious. During deep sleep, with the metabolic rate for oxygen and glucose falling by as much as 40% in some cortical regions, the cell's own cleanup systems can finally work without competition. The proteasome, a molecular machine present in every neuron, built from a ring of protein-degrading enzymes stacked into a hollow barrel, recognizes and dismantles damaged or misfolded proteins with a precision that feels almost deliberate, though it has no awareness of anything it encounters. During the low-energy hours of deep sleep, the proteasome's work can proceed against a smaller incoming tide of new metabolic damage. Autophagy, the process by which cells engulf and digest their own worn components, also runs at elevated rates during the brain's quiet hours, turning used organelles back into raw molecular material the cell can reuse. These are housekeeping functions, not glamorous, not visible from outside the cell, not experienced as anything by the sleeping person whose neurons are being slowly refurbished from within. Mitochondria, the dense, folded structures inside each neuron that carry out the work of energy generation, are among the most vulnerable components of the waking brain. They operate under high oxidative stress, generating the reactive molecules that gradually damage their own membranes and proteins. During deep sleep, when the demand for energy drops and the reactive load decreases, the conditions for mitochondrial maintenance improve. Damaged mitochondria are tagged, isolated, and broken down. Healthy ones proceed through their own replication cycle, gradually replacing what has worn out. By morning, the mitochondrial population in the cortex is in a small but real way in better condition than it was the night before. Astrocytes, the star-shaped supporting cells that outnumber neurons, and provide them with structural and metabolic support, are replenishing their stores of glycogen during the sleeping hours. Glycogen is the brain's local reserve of glucose, held in astrocytic granules and available to neurons in moments when blood glucose delivery is briefly insufficient. It is consumed across the waking day in proportion to neural activity and cognitive demand, depleted slowly but measurably. During deep sleep, when the neurons it supports have reduced their firing rates and their energy requirements, astrocytic glycogen replenishes, the reserves restored to a level ready to sustain the next day's activity. The sleeping brain is not depleting its fuel reserve, it is filling it back up. The immune system does not stand apart from the night's work. Deep NREM sleep is accompanied by elevated secretion of several signaling molecules, interleukin 1 beta and tumor necrosis factor alpha among them, that serve a double function. They promote sleep directly, acting on the brain's own circuits to deepen and extend slow-wave activity, and they also mobilize the immune response, directing the movement of immune cells and the intensity of their activity. The same molecules that call the body into its deepest rest are also the ones that marshal its defenses. Deep sleep and immune function are not parallel systems running alongside each other in the night. They are coupled, each driving the other. The depth of sleep and the depth of the immune response rising together across the recovery hours. Natural killer cells, a class of immune cell that circulates through the body's lymphatic system, identifying and eliminating cells that have been infected by viruses or that have undergone the molecular changes that precede cancer, show heightened activity during the sleeping hours. Their capacity for recognizing compromised cells, reaching its nightly peak during deep NREM. They move slowly through the lymphatic tissue, unhurried, in the dark of the body's interior. They do not know what they are looking for in any purposeful sense. They carry receptor molecules whose shape happens to fit the molecular signatures of infected and abnormal cells, and when the fit occurs, the response follows automatically, a lock finding its key in the dark. A 2019 study found that sleep promotes the activation of integrins proteins on the surface of T cells, the immune system's adaptive responders, that allow those cells to adhere more tightly to the antigen-presenting cells that carry information about what threats have been encountered. The night, in other words, is when the immune system does its paperwork. T cells physically bind to the cells that carry the day's threat signatures, consolidating information about what was encountered and preparing a more specific response. When sleep is shortened or fragmented, this adhesion is reduced. The briefing happens incompletely. The immune system meets the next day, slightly less informed than it might have been. The body's temperature drops slowly during deep sleep. Core temperature reaches its low point in the early hours of the morning, several degrees below its waking level, and this cooling is not incidental. It is actively driven by the hypothalamus, which coordinates the dilation of blood vessels near the skin's surface to radiate heat outward, reducing the metabolic cost of maintaining the deep sleep state and creating the thermodynamic conditions in which certain repair processes run more efficiently. Cooler tissue generates less oxidative stress. Proteins that are prone to misfolding at higher temperatures fold more reliably when the cellular environment is cooler and calmer. The temperature drop is part of the restorative architecture, not a side effect of inactivity. Across the neocortex, over the course of the night's deep sleep, a process of synaptic adjustment is taking place. Quiet, selective, operating at the scale of individual connections between neurons rather than at the scale of whole circuits. During waking, experience strengthens synapses. Every new thing learned, every stimulus noticed, every pattern recognized produces small but real increases in the efficacy of the synaptic connections involved. More receptor proteins inserted into the postsynaptic membrane, more neurotransmitter released with each signal, the connection itself made incrementally more responsive. This is how learning works at its most basic level. Experience changes the physical relationship between neurons, and the change persists. By the end of a full waking day, those changes have accumulated across billions of synaptic connections throughout the cortex. The brain is, in a measurable molecular sense, slightly more potentiated than it was at waking. The AMPA receptor subunit, known as GLUIL1, shows elevated phosphorylation, a chemical modification that increases synaptic efficacy at the end of the waking period, and the levels of this modification decline across the sleep period in parallel with the progress of slow-wave activity. The slow waves themselves, in their broad and synchronized sweeps, are thought to carry out this downward adjustment, reducing synaptic weights across the cortex gradually, selectively, preserving the relative differences in connection strength while reducing the overall level. The idea is not that connections are erased, it is that they are normalized. The brain ends the waking day slightly saturated, too many connections strengthened, the signal-to-noise ratio across the cortex gradually degrading as everything becomes slightly more excitable, slightly harder to distinguish from everything else. Deep sleep, in this view, is the edit, not a deletion, but a recalibration, the cortex brought back to a dynamic range from which tomorrow's experience can again register as something new. Molecular markers of synaptic potentiation peak at the end of waking and fall through the night. By morning, the cortex is quieter, and in that quiet, more capable of being changed again. The slow oscillations that sweep through the sleeping cortex are not passive. They are not simply the brain idling in the absence of input. Each slow wave is a moment of coordinated neuronal firing, the up state, followed by a moment of nearly complete neuronal silence, the down state, and the transition between the two is driven by the same synaptic activity the wave is also modifying. When neurons fire together during the up state, the connections between them undergo the same rapid strengthening that occurs during waking learning. When the down state arrives and activity ceases, those recently potentiated connections begin the process of normalization. The slow wave is both the agent and the result of the synaptic adjustment it performs. The brain's deep sleep is not separate from its plasticity. It is where plasticity is completed. This process happens differently in different regions of the cortex. Areas that were more active during the preceding day, more engaged by experience, more extensively modified by learning, show greater slow-wave activity during the night. The sleep EAG is in this sense a kind of map of the day's experience. The regions that worked hardest rest most deeply. If a task is learned that engages the motor cortex in the right hemisphere, the slow-wave power over that hemisphere increases during the following night. The sleeping brain is not uniform in its rest. It is organized by what has happened to it, and its deepest restoration is distributed exactly where it is needed. The pituitary gland rests again after its early night pulse of growth hormone, but the bloodstream continues to carry the signal outward. In skeletal muscle, cells called satellite cells, muscle stem cells that remain dormant during the waking hours, are activated by growth hormone's arrival and begin the slow process of repairing the microstructural damage that accumulates with physical use. The tiny tears and muscle fibers caused by exertion are addressed not during the effort, but during this quiet interval, when the biochemical conditions created by deep sleep allow the repair process to proceed without competition from the metabolic demands of movement. The soreness that follows a day of physical work begins to diminish before morning in increments too small to feel, while the person who worked and rested is entirely unconscious of either the damage or its repair. In bone, the same pulse has activated osteoblasts, the cells responsible for depositing new mineral matrix, which proceed through their slow cycle of construction across the night. Bone is not static, it is continuously demolished and rebuilt, old tissue cleared away by cells that dissolve mineral and organic matrix alike, new tissue laid down by cells that follow behind. The balance between the two populations shifts across sleep and waking, with the constructive side of this cycle reaching its peak during the hours of deep NREM. For a sleeping child, this process supports the physical elongation of bones across the years of growth. For a sleeping adult, it maintains the density and architecture of a skeleton that must last for decades. The liver, working in the metabolic context created by growth hormone and the other signals of deep sleep, shifts the body's glucose management toward a lower demand equilibrium. Blood glucose falls gradually across the sleeping hours, held within a narrow range by a careful hormonal balance. The pancreas reduces its insulin secretion as the metabolic rate drops. Fat cells release stored fatty acids into the bloodstream at a rate calibrated to the body's reduced but ongoing energy requirements. The entire metabolic system settles into a slower rhythm, not stopped, not suspended, but redistributed away from the immediate demands of waking activity and toward the longer-term work of repair. The kidneys slow their filtration rate slightly during deep sleep, reducing the production of urine and concentrating it, extending the body's fluid balance across the hours without requiring waking to drink. The blood pressure falls, the heart rate slows, the smooth muscle of the arterial walls relaxes, the cardiovascular system, like every other system, entering a lower demand state that allows the repair processes that depend on reduced mechanical stress to proceed. The arteries themselves benefit from the nightly reduction in pressure. The endothelium, the single-cell lining of the vessel walls, whose health is central to cardiovascular function across a lifetime, carries out its own maintenance during these hours, replenishing the molecular signals that keep the vessel wall flexible and resistant to the accumulation of inflammatory deposits. Outside the specific tissues being repaired, across the whole extent of the sleeping body, the autonomic nervous system has shifted its balance. The parasympathetic branch, the one associated with rest, digestion, and maintenance, holds a greater influence during deep sleep than at any other time of the 24-hour cycle. The sympathetic branch, which carries waking alertness, arousal, and the readiness for physical response, has pulled back. The ratio between them shifts measurably in the heart's rhythm. The intervals between beats lengthen and become more variable, a pattern called high heart rate variability that is associated with good cardiovascular health and the parasympathetic predominance of deep rest. Each heartbeat during deep slow-wave sleep is slightly more widely spaced than the one before it, the heart settling into its slowest nightly pace. The experience of the sleeping person through all of this is nothing. No awareness of the fluid moving through the brain's channels, no sense of the hormone pulse dispersing through the blood, no felt knowledge of the synapses being quietly adjusted across the cortex or the mitochondria undergoing their slow refurbishment inside each neuron, or the T cells pressing against the antigen-presenting cells in the lymphatic tissue of the neck and chest. None of it is experienced because the mechanisms of experience have been temporarily withdrawn. The thalamus has closed its gates. The cortex has reduced its effective connectivity to near nothing. The sleeping brain is as far from waking awareness as it will ever be, and in that distance, in that profound removal from the surface of things, the body's most essential work is carried out in full. This is not restoration that would happen anyway, regardless of sleep. It is restoration that requires sleep, specifically the deep, slow, electrically distinctive state of NREM stage 3, to proceed at the rate and depth that keeps a biological system viable across years and decades. When sleep is shortened, or when deep slow wave sleep is specifically disrupted, the markers of restoration are correspondingly reduced. Growth hormone secretion falls, glymphatic clearance is impaired, synaptic downscaling is incomplete, immune cell activity is reduced. The morning body is slightly less restored than it would have been. The deficit is small on any single night and is not experienced as anything specific. A little more fatigue, perhaps, a little more difficulty with demanding tasks, but it accumulates, and the accumulation has real consequences across the span of a life. There is something precise and unostentatious about the body's approach to all of this. No part of it announces itself. No signal rises to awareness to say that the cleaning is running, that the repairs are underway, that the immune system is conducting its nightly review. The whole vast coordinated enterprise of biological restoration proceeds in the absence of any witness, driven not by intention or awareness, but by the accumulated conditions of chemistry and physiology that deep sleep creates and sustains. The slow waves roll through the cortex. The fluid moves through the channels. The hormone disperses into the blood. The synapses are quieted and adjusted. The astrocytes, those star-shaped supporting cells distributed through every region of the brain, complete the refilling of their glycogen stores before the first light of the following day. Each granule of stored glucose represents a small reserve, a few seconds of support for the neurons nearby in moments of high demand. Across the brain, billions of such granules are restored across the sleeping hours, filled from the glucose that the blood delivers at a steady rate, even while the neuron's own demand has fallen. By the time the first slow-wave episode of the night gives way to a lighter stage, and the brain begins its gradual climb back toward a shallower sleep, the energy stores of the cortex's supporting tissue are quietly, precisely, completely restored. The night continues. The slow waves that characterize the deepest hours have done their work in the first two cycles, concentrated there by a biological economy that places the most intensive restoration at the beginning of the night when the accumulated need is greatest. Across the later hours, the character of sleep shifts, the deep valleys in the night's architecture growing shallower, the slow-wave episodes shorter, the balance gradually tilting toward other stages with their own distinct functions. But the restoration carried out in those first deep hours is already complete. The cleaning current has run, the hormone pulse has dispersed, the synaptic adjustment has proceeded through its first and most important pass. The sleeping brain does not know morning is coming. It keeps no clock the way a waking mind tracks time, but the physiology obeys its own schedule, and the schedule is ancient, older than language, older than the cortex that generates the slow waves, older than the particular configuration of proteins and channels and circuits that carries it out in any individual alive today. The pattern was present in the ancestors of mammals, adapted and refined across the long span of evolutionary time into the precise and layered system that runs every night behind the sealed gates of a sleeping skull. In the lymphatic tissue along the neck, the T cells that spent the first hours of the night pressing against antigen-presenting surfaces begin to circulate again. Their briefing is done. The integrins that held them in place across the slow wave hours have reduced their adhesion, releasing the cells back into the bloodstream to carry the information they've acquired toward whatever encounter the coming day may bring. The immune system is, in a small but real way, more prepared than it was yesterday. The preparations were completed in the dark, without witness, as all the most essential maintenance is. The astrocytic glycogen is full. The proteasomes have processed their load. The mitochondria that were most worn have been tagged and set aside for replacement. The growth hormone has reached its peripheral targets and set the synthesis machinery in motion. The slow waves, in their 10,000 passes across the sleeping cortex, have made their adjustments to the 10 billion connections that carry the brain's record of the world. Nothing is announced, nothing is commemorated. The work is simply done, the way a tide completes its cycle without any awareness that it has cycled. The water returning to the shore, the sediment settling, the surface smoothing, and the body of water resting in the same condition it was in before the activity began, ready to receive what the next tide brings. The glycogen stores are full. The brain, in the hours before it wakes, rests against its own completeness, quiet, restored, sealed from the world, carrying everything forward into the morning that is still some time away. In the hippocampus, deep inside the brain of a person who has been asleep for perhaps an hour, something barely visible on any recording instrument briefly ignites. It lasts less than a fifth of a second. Within that narrow window, shorter than the time it takes to blink, shorter than the gap between two heartbeats, a sequence of cells fires in rapid cascading order, each one activating the next, the way a line of stones falls, in a careful, practiced sequence. The pattern these cells trace is not random. It is a route, an afternoon walk, a face at a doorway, the particular angle of late light across a familiar surface. The cells that fired while that moment was first lived now fire again, in the same order, compressed to a fraction of the original time. The memory of something that once moved slowly through the world is rehearsed in the sleeping brain 20 times faster than it was ever actually lived. This brief event, high frequency, transient, gone almost as soon as it begins, is called a sharp wave ripple. It carries within its short electrical life the architecture of waking experience, replayed and compressed and delivered inward toward the slower rhythms that surround it. The cells that generate it belong to the deepest memory structure the brain possesses, a curved formation sitting beneath the temporal lobe on each side, ancient in evolutionary terms, and extraordinarily sensitive to the geography of experience. In sleep, these cells do not merely rest, they work quietly, repeatedly, across the long hours of the night, rehearsing what the day deposited and passing it, piece by piece, to the wider cortex that surrounds them. What surrounds them during deep NREM sleep is not quiet in the ordinary sense. The cortex is moving in slow, enormous waves, the great rolling oscillations that define this stage, vast in amplitude, glacial in pace, sweeping from the front of the brain toward the back in a rhythm of roughly 1 cycle per second, or slower. Within each of these waves, as its rising slope builds and the cortex enters the brief active period of strong neural firing, something else appears. From the thalamus, the deep relay structure beneath the cortex, a short burst of faster oscillations arrives, 12 to 15 cycles per second, lasting perhaps a second or two at most, and nestles within the upslope of the cortical wave, like a bird settling briefly into a current of warm air. This faster burst is a sleep spindle. Its arrival is not accidental. The timing is precise, and the precision matters enormously. The 3 rhythms, the slow cortical oscillation rolling at under 1 cycle per second, the sleep spindle nested within its rising phase, and the hippocampal sharp wave ripple fired within the narrow excitable window the spindle creates, are not simply happening in parallel. They are synchronized, they are coupled, and the coupling between them is the mechanism through which the sleeping brain takes what the waking day deposited in the hippocampus and begins the long, careful work of moving it somewhere more permanent. The hippocampus is suited to initial storage. It is fast, associative, and capacious in the short term. A new face, a new fact, the layout of an unfamiliar street, these are captured there quickly and held with reasonable fidelity for hours or days. But the hippocampus was not designed to be a permanent archive. It is more like a working surface than a filing cabinet, necessary for the first draft, but not the final home. The neocortex, the vast folded sheet of gray matter wrapped around the outside of the brain, is the final home. It holds memories that have been abstracted from their original context, woven into existing knowledge, and stabilized against the interference of future experience. The transfer from one structure to the other is slow. It takes nights, possibly many of them. The slow oscillation is the mechanism of transfer. During its rising up state, when cortical excitability briefly peaks and neurons fire in near unison, the hippocampus has a narrow opening through which its sharp wave ripples can influence cortical synapses. The spindle, arriving precisely at that moment, holds the excitability window open a little longer and amplifies the channel between the two structures. When the hippocampal ripple fires during this combined window, the upstate and the spindle simultaneously present. The signals it carries reach cortical synapses in a state of heightened plasticity, which means the connections they touch are more likely to be modified, more likely to retain some trace of what passed through them. The memory is not transferred all at once like copying a file from one location to another. It is laid down gradually across many repetitions, each replay leaving a slightly deeper groove in the cortical tissue it touches. The evidence for this process arrives from many directions, accumulated across decades of sleep research, and, more recently, from carefully designed experiments in which the timing and content of memory can be traced before and after a night of sleep. Paired associate learning experiments offer one clean window into the system. A person studies a set of word pairs, arbitrary pairings, matched by a researcher rather than by any natural logic, and is tested on recall, either after a night of sleep or after an equivalent period of wakefulness. Across many such experiments, the results are consistent enough to be trusted. Sleep produces 20 to 40% better recall than waking rest under conditions otherwise matched. The benefit is not distributed evenly across all types of sleep. It concentrates in the slow wave episodes. The more slow wave activity across the night, and the higher the density and amplitude of sleep spindles nested within that activity, the stronger the recall on the following morning. 20 to 40% is a large difference. It is not a marginal effect visible only in statistical aggregates. It is a difference that can be felt in the ordinary experience of learning anything that requires repeated encoding, a language, a set of procedures, a body of facts whose connections matter as much as their individual content. The night between the first study session and the next morning's test is not an interruption of the learning process. It is a continuation of it, running in a different medium, through different mechanisms, while the person who is learning is entirely unaware that any work is being done. The kind of memory most consistently supported by deep NREM sleep belongs to the category broadly described as declarative, the explicit, conscious memory for facts and events that can be called to mind deliberately and narrated in language. What year something happened. The capital of a country studied in childhood. The plot of a book read years ago. The names of people who were once known well. These are the memories that fade most visibly across a life, and they are also the memories whose consolidation depends most directly on the hippocampal neocortical transfer that slow-wave sleep enables. When slow-wave sleep is disrupted by noise, by alcohol, by certain medications that suppress deep sleep without eliminating it entirely, declarative memory suffers measurably on the following day. The facts are harder to retrieve. The connections between them are less reliable. The material that was studied the previous evening feels less firmly held. The memory for movement and skill belongs to a somewhat different system and follows a somewhat different consolidation path. A finger-tapping sequence, a balance task, the particular muscular coordination required by a new instrument or a new sport. These are held in a network of structures that includes the motor cortex, the basal ganglia, and the cerebellum, and their consolidation across sleep involves a different weighting of sleep stages. Yet slow-wave sleep contributes here too, not primarily as the direct vehicle of motor memory encoding, but as something more like a protective mechanism. Across the night, the slow oscillations and their associated synaptic downscaling create conditions in the motor cortex that prevent newly formed representations from interfering with the skills already consolidated there. The night sleep does not simply add the new skill to the existing store, it edits the entire landscape, lowering the general level of synaptic excitation in a way that stabilizes the old while making room for the new. The morning's improved fluency on a practice sequence is partly the result of a night spent not just sleeping, but managing the architecture of what is already known. This editing function, the selective reduction of synaptic weights across the cortex during slow-wave sleep, is the central claim of a major theoretical framework in sleep research. The hypothesis, developed by Giulio Tononi and Chiara Cirelli over many years, proposes that waking life produces a net increase in synaptic strength across the brain. Every new experience, every learned association, every sustained period of attention leaves behind a slightly potentiated synapse, a connection slightly stronger than it was before. This accumulation is useful up to a point, but the brain cannot sustain indefinitely increasing synaptic weights. The metabolic cost rises, the signal-to-noise ratio falls. The ability to distinguish the strong, meaningful connections from the weaker background noise begins to degrade. Slow-wave sleep on this account exists in part to renormalize the system, to bring synaptic weights back down toward a sustainable baseline, preserving the relative differences that encode what was learned while reducing the absolute level of excitation across the network. The molecular evidence fits the picture. The phosphorylation state of particular receptor proteins at cortical synapses, markers of how recently and how strongly those synapses have been potentiated, peaks at the end of a normal waking day and declines across the sleep period, in close parallel with the rise and fall of slow-wave activity. As the night progresses and slow-wave activity accumulates, the molecular signatures of synaptic potentiation recede. By morning, the synaptic landscape across the cortex has been quietly revised, not erased, but edited, made more efficient, the most strongly potentiated connections preserved, and the weakest pruned back towards silence. The memories that survive this editing are not necessarily the most recent or the most frequently rehearsed. They are the ones whose underlying synaptic traces were strong enough to endure the night's calibration. Not all memories survive equally, and the factors that determine which ones endure are not entirely understood. What is known is that emotional salience plays a role, that prior knowledge creates a scaffold that makes new information easier to integrate and therefore more likely to persist, and that attention during encoding matters. The depth with which something was processed in the first place shapes what the sleeping brain has to work with. Sleep consolidation does not rescue poorly encoded material. It amplifies and stabilizes what was already there, and it does so most effectively when the waking encoding was deliberate and engaged. There is a class of experiments that makes this visible in a way that would seem improbable if the results were not consistent across many laboratories. While a person sleeps in a stage of deep slow wave activity, a faint cue is played into the room, a sound or a scent associated with a specific item that was studied during the previous waking session. The cue is far below the level that would wake the sleeper. The EEG trace does not show any obvious arousal, yet the response in the sleeping brain is measurable and specific. A brief increase in slow oscillation power, a momentary pulse of hippocampal activation, a targeted surge in consolidation activity oriented toward the memory associated with that cue. In the morning, the cued memories show better recall than the uncued ones, not dramatically but reliably and selectively. The sleeping brain can be steered gently and from the outside towards specific material, and it will preferentially consolidate what it has been pointed toward. This technique, known as targeted memory reactivation, has been extended well beyond the laboratory demonstration of the principle. Researchers have used it to strengthen the retention of foreign vocabulary learned before sleep. Others have applied it to motor skills, replaying the tones associated with a finger tapping sequence during subsequent slow-wave sleep and finding improved performance in the morning. More recently, work has explored whether the same approach can be used to weaken rather than strengthen particular associations, presenting cues linked to implicit bias measures during sleep and finding modest but measurable reductions in bias strength on subsequent testing. The sleeping brain, it turns out, is not indiscriminately absorbing everything that reaches it. It is selectively responsive, especially to material linked to slow-wave oscillatory activity, and especially during the deep NREM episodes concentrated in the early hours of the night. That selectivity has a geography. The cortical regions most active in the consolidation of a particular kind of memory are the ones whose slow oscillations are most directly involved in the process. For memory with a strong spatial or contextual component, the areas involved in processing the original experience are engaged again during slow-wave sleep, their oscillatory activity tracking the specific content being consolidated. For language-based declarative memories, frontal and temporal regions show the relevant slow-wave involvement. The slow oscillation is not a uniform signal washing over the entire cortex simultaneously. It travels, it propagates. And where it propagates most strongly is shaped at least in part by what the waking brain spent the day doing. The propagation itself is worth holding in the mind for a moment. The slow oscillation begins most often near the front of the cortex, in the regions most associated with planning, attention, and the top-down organization of thought, and it spreads from there toward the back, sweeping across the surface over the course of roughly half a second. As each region is touched, it passes briefly into the upstate, and then drops into silence, the population of neurons there rising and falling together in the great collective rhythm of deep sleep. The wave is not a single event, but a repeating pattern, cycling through at below 1 cycle per second across the hours of early sleep, each iteration another opportunity for hippocampal signals to reach the cortex at the precise moment of maximal excitability. The repetition is the mechanism. No single cycle is sufficient. It is the accumulated effect of hundreds of cycles across the course of a full night's deep sleep that gradually transfers the memory from its initial temporary store to something more durable and more widely distributed. The neural signature of this transfer is visible if one looks for it carefully. An fMRI scan, taken while a subject recalls a newly learned fact on the morning after sleep, shows a different activation pattern than one taken immediately after the original learning. The hippocampus is less engaged at retrieval. The neocortical regions are more engaged. The memory has moved, not all at once and not completely, but measurably, from a structure suited to rapid encoding toward a network suited to long-term storage. The same fact, held in a different architecture, requiring a different neural path to retrieve. This shift takes more than a single night. For complex material with many internal connections and dependencies, the full consolidation process extends across multiple sleep episodes. But each night of adequate slow-wave sleep advances it. The concept of consolidation is sometimes imagined as a simple process of strengthening, as if memory were a stone being polished to a brighter surface by the night's attention. The reality is considerably less tidy and in some ways more interesting. Consolidation involves not only the stabilization of what was encoded, but also the reorganization of how it is represented. Material that was memorized as a list of disconnected facts may be retrieved the following morning as something more structured, the relationships between items clearer, the connections more intuitive, the underlying patterns more accessible. Sleep appears to do something to the knowledge that goes beyond preservation. It transforms it at least partially from a set of discrete encoded items into something more integrated, a representation that has been fitted, at a level below deliberate awareness, into the web of what was already known. Some of this integration arrives in a form that can be tested directly. Problems that require the discovery of a hidden rule, where the rule is never explicitly taught but must be inferred from patterns in the training data, are solved more readily after sleep than after equivalent wakefulness. The insight, when it comes, feels sudden and intuitive rather than effortful. But the groundwork for it was laid during the slow-wave episodes of the previous night, when the encoded material was replayed and reorganized and fitted into the broader structure of existing knowledge in a way that made the hidden pattern accessible to the waking mind. Sleep does not only preserve what was learned, it sometimes reveals what was already there to be found. The hippocampus across the night does not replay all memories equally. It selects. The mechanism of selection is not fully understood, but several factors are known to play a role. Novelty matters. Memories of experiences that were new and unexpected are more likely to be reactivated. Emotional tone matters. Memories tagged with strong feeling are prioritized. Relevance to prior goals matters, though how the hippocampus knows what was relevant is a question whose answer involves structures well beyond it. It is an edited version, shaped by filters that are biological in their mechanism, but biographical in their effect, reflecting something about what the sleeping brain judges worth preserving. Across the first two cycles of the night, when slow-wave sleep is at its longest and most intense, the rate of memory consolidation is highest. The hippocampal neocortical dialogue is most active, the sharp wave ripples most frequent, the slow oscillations deepest and most consistently propagating. As the night advances and slow-wave episodes shorten, the balance of sleep stages shifts, the deep NREM giving way to longer REM periods, with their own distinct contributions to memory processing that operate through different mechanisms and on different memory systems. The early night and the late night do different work. Neither is dispensable, but the foundation, the initial transfer of declarative material from hippocampus to neocortex, the stabilization of newly formed representations against interference, the global recalibration of synaptic weights, all of this is concentrated in the early, deep, slow wave-rich episodes of the night. A person who loses the early hours of sleep through a late bedtime loses disproportionately more slow-wave sleep than total sleep. The slow-wave episodes are front-loaded. They appear first, and if the first two cycles of the night are cut short, the consolidation work of those cycles is lost and cannot be fully made up by the lighter sleep that follows. This is not a marginal consideration. For material that was studied the previous evening, the difference between sleeping at an ordinary hour and losing the first 2 hours of sleep represents a difference in the depth and completeness of overnight consolidation that is visible in subsequent recall performance. The timing of sleep matters, not only its duration, against the broader backdrop of what slow-wave sleep does to memory. What is most quietly striking is perhaps not the magnitude of the effect, but its consistency. Night after night, without instruction, without awareness, without any effort on the part of the sleeper, the brain conducts its own careful editorial review of the day's experience. The review is not random. It is guided by the architecture of the slow oscillation, shaped by the precision of the spindle's timing, and coordinated through the narrow windows of hippocampal excitability that the two rhythms together create. The memories that emerge from the process are not identical to the memories that entered it. They are different in small but important ways, more stable, more integrated, better fitted to the knowledge that preceded them. This quiet editorial work is most concentrated in the small hours before midnight and just after, the deep valley of early sleep that the sleeping body falls into without ceremony and climbs out of so gradually that the transition is rarely noticed. During those hours, the great slow waves roll through the cortex in their steady, ancient rhythm, each one carrying within its rising phase the brief, bright synchrony of a spindle, each spindle briefly opening the channel through which the hippocampus speaks to the neocortex, each ripple firing its compressed replay of an afternoon's accumulated experience into that brief open window. The memory of a face, a rote, a fact, a word learned in a foreign language, all of these pass through that channel again and again across the long unhurried depth of early sleep. In the hippocampus, a sharp wave ripple fires. It lasts 1/10 of a second. Within it, the memory of something lived is rehearsed one more time, compressed, rapid, exact, and the signal it carries travels outward through the coupling of 3 rhythms toward a cortex that is ready, briefly, to receive it. The slow oscillation rolls on, the spindle fades, the ripple ends, and somewhere in the neocortical tissue that was touched by that brief exchange, a synapse is infinitesimally altered. The trace of today's experience laid down a little more deeply into the permanent fabric of what is known. The process will repeat. It has been repeating since shortly after sleep began, and it will continue until the night's slow wave episodes are spent and the brain tips into a lighter, faster mode. The memories accumulate their changes across the night, quietly, without the sleeper's knowledge, without any sensation of work being done. By the time morning approaches and the body begins its slow return toward wakefulness, what was learned the previous day has been moved, at least partially, from the temporary holding of the hippocampus into the wider, more durable architecture of the neocortex. The neural path to retrieval is different now. The representation is more stable, less vulnerable to interference, more thoroughly integrated with what came before it. Something that was known loosely the evening before is known a little more firmly by morning, not because of any additional exposure, but because the sleeping brain spent the night quietly doing what it was built to do, in the deep, slow electrical silence of its most restful hours. The slow oscillation that carried this work has already moved on. It swept from the frontal cortex toward the back, through its upstate and its downstate, and it is gone, replaced by the next cycle and the one after that, each rolling through the sleeping brain with the same unhurried amplitude, the same steady rhythm, the same capacity to carry the night's careful work forward. The ripples that fired within those cycles have long since ended, their microsecond lives complete. What remains is not the electrical event itself, but what the event accomplished. A synapse altered by a fraction of a millivolt, a connection made fractionally more durable, a memory settled a little more deeply into the permanent landscape of the mind. Outside the window, the night is still, the air is cool, and the city, wherever there is one, has gone quiet in its own way. Not silent, but softer. The daytime layers peeled back to something slower and lower. Inside a room somewhere in that quiet, a person lies in the first deep valley of the night, unreachable by ordinary sound, their brain moving in its great slow waves, the frontal lobes rising and falling, the signal traveling backward across the cortex, the hippocampus firing its brief, precise replays into the coupling of rhythms that holds the whole system together. The sleep is deep, and the work is steady, and the night, for the moment, is exactly long enough. Deep inside the thalamus, in the densest hours of the early night, a thin, curved shell of neurons has drawn the darkness in around the sleeping brain and sealed it there. The shell is not large. It wraps the thalamic core the way bark wraps a young branch, close, continuous, its cells pressed against the relay tissue beneath them, in a layer only a few neurons deep. Nothing about its appearance suggests power. Everything about its activity does. These neurons are inhibitory. Their transmitter is a small molecule that quiets whatever it touches, and during the deepest hours of slow-wave sleep, they release it in slow, sustained waves against the relay cells just below them. The relay cells fall silent. The signals arriving from the ears and the skin and the eyes, signals that would, in any waking moment, climb upward through the thalamus toward the cortex, now arrive at a gate that does not open. They travel to the gate, they stop there. The cortex above, waiting in the dark, receives nothing. This structure, the thalamic reticular nucleus, does not act alone, and it did not arrive at this state without reason. The molecules that keep the brain alert during waking hours, the ones released in fine continuous threads by clusters of cells in the brainstem and the hypothalamus, have by now withdrawn almost entirely. Norepinephrine, which narrows attention and lifts the arousal of everything it touches, has fallen to its lowest concentration of the 24-hour cycle. Acetylcholine, which floods the cortex during waking and brings its circuits into sharp and rapid engagement, has receded. Histamine, which keeps the thalamus tuned and responsive, has quieted. In their absence, the reticular nucleus operates without the braking force those signals provide when the body is awake. It fires more fully. Its inhibitory grip on the relay cells beneath it deepens. The gate, already present in lighter sleep, becomes something closer to a wall. The sleeping brain has not gone dark. The cortex above the sealed thalamus is alive with its own slow rhythms, generating waves so large they can be measured through bone and skin and the gel of a conductive electrode pressed gently against the skull. But it is generating those rhythms from within itself, from the interaction of its own cells in their slow alternating cycle of firing and silence, and not from anything the world is sending up from below. The outside world is still out there. It continues. The sounds of the house, the small shifts in temperature, whatever moves beyond the window in the earliest darkness of the night, all of it persists, traveling through air and bone and the conductive tissue of the ear. It arrives, and the thalamus, its reticular neurons firing steadily in their slow inhibitory waves, absorbs it all and carries it no further. This is not passivity. It is one of the most active things the sleeping brain does all night. The inhibitory transmitter at the center of this ceiling, gamma-aminobutyric acid, the brain's most abundant quieting chemical, acts here through a specific class of receptor that produces a long, slow, hyperpolarizing current in the cells it touches. Where a faster receptor would open and close in milliseconds, this one keeps its channel open for hundreds of milliseconds at a time, holding the relay neurons below, resting potential for long stretches, making them resistant not just to the ordinary signals arriving from the senses, but to almost any signal at all. Each firing from the reticular shell lays down this slow inhibition, like a hand pressing gently and steadily on a surface, rather than striking it once and lifting. The pressure accumulates. The relay cells stay down. Below the thalamus in the brainstem, the neurons that would normally release the waking molecules continue to rest. The slow oscillation in the cortex above continues its long cycle, neurons rising together into the up state, firing briefly in near unison, then releasing into the deep silence of the down state, the whole population hyperpolarized and still for a fraction of a second, that in this condition feels, from inside the sleeping brain, like nothing at all. The wave sweeps forward across the cortex from the front of the head toward the back, traveling at a speed that takes roughly half a second to cross the full extent of the sleeping brain. It arrives at the posterior cortex. It subsides. Another begins from the front again, slow and large and regular, the rhythm of a brain that is turned entirely inward. A tone sounds in the room. And in lighter sleep, even a soft sound can reach the auditory cortex within a tenth of a second, producing a deflection in the EEG record, a response, measurable, the brain acknowledging what it has received. In this depth, something different happens. The sound travels through the air and enters the ear. The hair cells of the cochlea respond to it in their usual way, converting the pressure wave into electrical signals that travel along the auditory nerve toward the brainstem. The signal climbs. It arrives at the thalamus. The medial geniculate nucleus, the thalamic way station for auditory information, receives it, and there, with the reticular nucleus pressing down its slow sustained inhibition onto the relay cells, the signal reaches the gate and stops. The auditory cortex, 90 milliseconds above the source, receives nothing, or so close to nothing that the EEG trace barely stirs, a faint, delayed, attenuated shadow of what the response would be in any lighter state, or no visible response at all. The sound happened. The ear registered it. The brain, in this depth, was not there to receive it. In lighter stage 2 sleep, a sound of sufficient intensity would produce a K-complex, a sharp upward deflection followed by a slower wave. The sleeping cortex's characteristic acknowledgement of an intrusion it has chosen, for now to absorb without waking. The K-complex is sometimes described as a protective response, the sleeping brain using a single organized wave to suppress the arousal that the sound would otherwise trigger. In slow-wave sleep, even this response fades. The cortex is running its own program, committed to its own slow oscillatory rhythm, and an incoming signal from the outside world is not easily strong enough to interrupt it. The threshold for arousal, the minimum intensity a stimulus must have to pull the sleeping brain back toward wakefulness, is higher here than at any other stage of the night. It rises not because the brain has stopped working, but because the work it is doing is protected by the depth of its own electrical activity. To understand how thoroughly the sleeping cortex has withdrawn from the world during slow-wave sleep, researchers developed a method that bypasses the thalamic gate entirely. A brief magnetic pulse, delivered through a coil held against the scalp, can induce a small electrical current in the cortex directly, not through the sensory pathways, not through the thalamus, but straight into the cortical tissue from outside the skull. In a waking brain, this kind of pulse produces a complex response that travels across the cortex for hundreds of milliseconds, activating one region and then another in a cascade of interaction that reflects the rich, far-reaching connectivity of the alert brain. The response is complicated. It spreads widely. It persists. The same pulse delivered during slow-wave sleep produces something entirely different. The cortex at the site of stimulation responds. It generates a large slow wave, a response that can be measured clearly at the electrode closest to the point of stimulation. But that wave does not travel. It does not propagate to distant cortical regions the way the waking response does. It appears at its origin, expands slightly, and then stops, absorbed into the surrounding silence as if the surrounding tissue were not connected to it at all. The rest of the brain does not receive the message. The measure developed to capture this, the perturbational complexity index, a quantification of how complex and widely distributed the cortical response to a direct perturbation turns out to be, falls during slow-wave sleep to its lowest value of any natural brain state. Wakefulness produces a high value. The response to perturbation is complex, widespread, informationally rich. REM sleep, in which the sleeping brain generates something close to the electrical activity of waking, produces a similarly high value. Deep NREM produces the lowest value measured outside of general anesthesia or the disorders of consciousness. The cortex during slow-wave sleep is not merely quiet. It is, in some precise and measurable sense, disconnected from itself. Each region has withdrawn into its own slow rhythm. The connections between them still exist. The white matter tracks that link frontal to parietal to temporal cortex are still in place, still structurally intact. But the slow oscillations downstate, the period of deep hyperpolarization, makes those connections functionally silent for the fraction of each cycle during which the neurons, on both ends of them, are not firing. The disconnection is not anatomical, it is electrical, rhythmic, recurring every second or so as the slow wave moves through its cycle. And in this rhythmically disconnected state, the perturbation of one region cannot ripple outward to become an experience, a perception, or an awareness of anything. The sleeping brain during slow-wave sleep is, in this sense, as close to a brain that is not processing experience as a living electrically active brain can be. The person lying in the room does not know any of this. There is nothing to know from inside it. The down state of the slow oscillation is a period of neural silence, so complete that it produces nothing, no sensation, no thought, no flicker of internal image or sound. It is a small gap in experience, recurring roughly once a second, and the upstates between them are not long or complex enough to support anything a waking mind would recognize as awareness. The person is present, the brain is active, and yet for the duration of the deep slow-wave episode, there is essentially no one home, not in the sense that would require arousal or attention, not in the sense that could be interrupted by the sounds of the ordinary world, not in any sense that the person themselves, waking hours later, will have any access to. They will remember none of the night's deep, slow wave sleep, not because something failed to record it, because there was, for those stretches, nothing to record. What happens then when the sleeper is forced out of this depth? The thalamic gate lifts. The reticular neurons relax their hold. The relay cells below them, freed from the sustained inhibitory pressure, begin to respond again to incoming signals. The cortex above starts receiving information from the outside world, the shake of a shoulder, the sound of an alarm, the gradual brightening of a room. The slow oscillation continues for a few cycles before it begins to break apart, its amplitude falling, its frequency rising slightly, the long sweeping waves giving way to faster, less organized activity. The eyes open, but opening the eyes and waking are not the same thing. Not here. The frontal EEG in a person forced awake from slow wave sleep continues to show delta range activity for minutes after they have opened their eyes and confirmed, verbally, that they are awake. The slow oscillatory residue persists in the frontal lobes, the same lobes that in waking support working memory, executive function, planning, and the flexible deployment of attention. While that residue is present, these functions are impaired. Reaction times are slower. The ability to hold several pieces of information in mind simultaneously is reduced. Decision-making is blunted in ways that are measurable on standard cognitive tasks and not subtle. The person answering a question may genuinely believe they are thinking clearly. The response latency, the error rate, and the quality of the answer tell a different story. This state has a name: sleep inertia, the dragging, thickened quality of cognition that follows forced arousal from the deepest sleep is most severe and most prolonged when the arousal comes from slow wave sleep rather than lighter NRE or REM. It can last 20 minutes. In some circumstances, particularly following significant sleep deprivation, it can last longer. It is not merely the feeling of tiredness, it is a genuine impairment. The frontal cortex still running residual slow oscillatory activity, while the brainstem and subcortical arousal systems have already signaled morning, the two systems briefly out of step with each other. The brain divided between the depth it was pulled from and the wakefulness it has been asked to perform. The body knows the difference. The brain knows the difference in whatever wordless way a brain can know anything at all. In the nights following sleep deprivation, whether from a single night of acute total loss or the accumulated shortfall of many nights of insufficient hours, the brain reclaims slow-wave sleep with a precision that reveals how carefully it tracks what it has been denied. The hypnogram on the first recovery night drops earlier and deeper than on any baseline night. The first N3 epoch begins sooner after sleep onset, lasts longer than it would on a rested night, and contains slow waves of higher amplitude, their peak-to-peak voltage elevated, the delta power across the frontal channels greater, the period of maximal disconnection from the outside world arriving with greater force, because the pressure that drives it has had longer to accumulate. What drives this accumulation is the waking brain itself. Every hour of consciousness, every act of attention and thought and sensory engagement produces changes in the synaptic landscape of the cortex, potentiated connections, strengthened weights, the molecular signatures of use. These changes build across the waking day in a way that is not directly felt as tiredness, at least not in the early hours, but that registers in the frontal EEG as a slow and steady rise in a particular marker, the homeostatic sleep pressure that will, when sleep finally comes, express itself as the depth and intensity of slow wave activity. The longer the waking period, the higher the marker rises. The deeper and longer the subsequent slow wave sleep must be to bring it back down. The relationship is proportional and consistent enough that the frontal delta power during slow wave sleep serves as a direct readout of how much waking experience preceded it. A brain that has been awake for 16 hours produces more delta power during its first slow wave episode than a brain that has been awake for 12. A brain deprived of sleep for 36 hours produces a recovery night of extraordinary slow wave intensity. The system does not forget. It does not average across days or discount the debt accumulated on short nights. It tracks with molecular fidelity what was spent and what is owed, and the slow wave sleep of the recovery night is the repayment, not approximate, not rounded, but precise. This homeostatic system runs alongside a separate clock, the circadian rhythm, driven by the suprachiasmatic nucleus in the hypothalamus, running on a cycle of just over 24 hours, synchronized to light and darkness, determines when the brain is willing to release into sleep and when it begins preparing for waking. The circadian rhythm and the homeostatic pressure operate independently, but they interact with exquisite coordination in the ordinary course of a well-slept life. The two systems reach their joint alignment for sleep in the late evening, when the circadian clock shifts toward night and the homeostatic pressure has been rising for most of the waking day, so that the conditions for deep and restorative sleep arrive together rather than separately. The result, across a full night, is the pattern visible in the hypnogram. The first 90-minute cycle plunges deepest. The slow wave episode within it is the longest of the night, its delta waves the highest in amplitude, its sensory threshold the most elevated, its disconnection from the outside world the most complete. The second cycle is slightly shallower, the third shallower still. By the early morning hours, the episodes of slow wave sleep have shortened, and in many adults have ceased entirely, replaced by the long REM periods that dominate the final cycles of the night. The homeostatic pressure falls across those early deep episodes and falls faster than it would during equivalent hours of wakefulness, because slow wave sleep discharges it more rapidly than lighter sleep. The brain is repaying its debt in large early installments, the deepest and most metabolically restorative episodes first, then shallower ones as the pressure eases. By morning, the debt is cleared. The cortex emerges from sleep with its synaptic weights adjusted, its metabolic resources replenished, its slow oscillation machinery wound down, and its readiness for the coming day rebuilt from the inside, quietly, completely, and without any of it being visible from outside the sleeping body. None of this required effort. None of it required intention. The thalamic gate opened and closed on its own schedule. The slow oscillations swept the cortex front to back at their own rhythm. The reticular neurons fired and relaxed and fired again, measuring nothing, intending nothing, simply doing what the chemistry of their membranes and the signals arriving from the brainstem made them do in the dark, and the sleeper lay still through all of it. There is something in the quality of the stillness that belongs specifically to slow wave sleep, distinct from the stillness of lighter stages. In stage 2 sleep, the body occasionally shifts, adjusting position, the limbs moving in small reflexive realignments. In REM sleep, the eyes move rapid, conjugate, unmistakable, while the body is held in the muscular paralysis that keeps the dreaming limbs from acting out the dreaming mind's intentions. But in the deep slow wave epochs of the first half of the night, the body settles into a stillness that is different in kind from both. Heart rate has slowed, blood pressure has fallen, the breathing has deepened and steadied into a cadence that matches, loosely, the slow oscillatory rhythm of the brain above it. not precisely coupled, but drawn toward the same low frequency, the same long, unhurried interval between one breath and the next. The muscles, no longer receiving the constant small corrective signals of waking motor tone, lie fully relaxed. The jaw slackens. The hands, which in lighter sleep might still carry a trace of the day's tension in the curl of the fingers, open slowly over the course of the deep sleep episode, the flexor tone releasing in increments as the slow wave activity deepens. If the sleeper is observed in this state, the face is composed in a way that waking faces rarely are, not the active blankness of effort, not the micro-tension of maintained attention, but something genuinely released, the muscular layer of expression given over entirely to gravity. The body is warm. The metabolic rate has fallen 20 to 40% lower than the waking baseline by the measures available to science, and the reduced heat production is offset by the redistribution of blood flow away from the surface of the skin, which conserves core temperature even as the peripheral tissues cool slightly to the touch. The hands away from the core are often cooler than they were an hour before sleep. The face retains more warmth. The core temperature itself, the deep internal temperature, measured in the great vessels and the organs, continues a slow fall that began at the start of sleep and will reach its lowest point somewhere in the middle of the night. The body's circadian rhythm and the metabolic quieting of deep sleep pulling it downward together. The room around the sleeper is ordinary. There is nothing remarkable about it. The darkness is the usual darkness of a bedroom with the curtains drawn, the silence, the ordinary silence of late night in a house where nothing is moving, the smell of sheets and wood, and the particular quality of recycled air in a closed room, not remarkable in any specific way, but familiar enough to be the condition of sleep itself, the background against which the brain produces its deepest work. The sleeper does not know any of this either. The ordinary room is as invisible to them as the extraordinary activity occurring 3 centimeters inside their skull. The thalamic reticular nucleus fires on. The relay cells below it stay quiet. The cortex continues its slow, self-organized rhythm, each wave traveling from the front of the head toward the back, each wave carrying hundreds of millions of neurons briefly into firing and then releasing them into silence. The silence lasts a little under a second. The next wave begins. In the ventromedial hypothalamus, cells that belong to the circuit regulating growth hormone release are receiving a signal that has been building since sleep onset. The first and largest pulse of growth hormone, the dominant release of the 24-hour cycle, arrived in the bloodstream within the first hour, or 90 minutes after sleep onset, timed to the first deep slow wave episode with a consistency that persists across decades of adult life. The hormone has been circulating since then, reaching the muscles and bones and organs, carrying the instruction that the repair work of the night may begin. In skeletal muscle, where the day's activity has left damaged contractile proteins and depleted energy stores, the growth hormone signal begins activating the anabolic processes that replace what was spent. In the liver, metabolic patterns shift. In connective tissue, the proteins of structure and elasticity are slowly turned over, the damaged molecules marked for degradation, the synthesis of replacements beginning in the slow hours of the deepest sleep. This is happening without announcement. The blood carries it silently. The cells receive it and respond. None of it produces a sensation. None of it will be remembered. The biological record of the night's first deep slow wave episode is being written not in the brain, but in the chemistry of the body's periphery, in the state of tissues that will be slightly different in the morning than they were at the start of sleep, repaired by increments too small to feel, restored by processes too quiet to name. Alongside this, in the white matter channels that run through the sleeping brain close to the walls of the blood vessels, a fluid moves slowly in the direction of flow. Cerebrospinal fluid, produced in the ventricles and circulating through the spaces of the brain and spinal cord, pushes through the perivascular channels during sleep in pulses that correspond to the low frequency oscillations of the sleeping brain, each slow wave associated with a small change in cerebrovascular tone that drives the fluid through its channels with slightly more force than during waking. The interstitial space through which this fluid moves, the narrow gaps between cells that in the waking brain are compressed and small, has expanded during sleep. The astrocytic cells that fill most of the brain's volume, having changed the geometry of their membranes in a way that widens the channels available for fluid flow. The metabolic residue of the day's thinking, small proteins, signaling molecules, the molecular byproducts of synaptic activity, moves from the interstitial space into the fluid and is carried outward toward the larger drainage pathways that will remove it from the brain entirely. The fluid moves slowly. The clearing is not fast, but across a full night of sleep, across the repeated deep slow wave episodes of the first half of the night in particular, the cumulative effect of this slow continuous clearing is substantial. The brain emerging from sleep with its interstitial chemistry, measurably different from its state at sleep onset. The concentrations of certain waste molecules lower, the conditions for the following day's neural activity slightly cleaner than they would have been without the night's work. All of this, the glymphatic clearing, the growth hormone pulse, the immune activity, the synaptic adjustment, the slow restoration of cellular machinery across the cortex, proceeds in the same silence as the thalamic gating, the same darkness as the reticular neuron's steady inhibitory discharge. It is not coordinated by a central controller. It is not supervised or checked against a plan. Each system responds to the conditions of deep, slow-wave sleep, the low metabolic rate, the low arousal, the particular hormonal and neuromodulatory environment of deep NREM, and proceeds according to its own logic, its own receptors and signals and downstream effectors, in the same space and the same hours as all the others. The organism restores itself the way a river restores its banks, not by design, but because the conditions that make restoration possible are present, and the processes that accomplish it need only time and the right quiet to do their work. Across the night, as the homeostatic pressure falls, the slow wave episodes grow shorter. The second N3 epoch is briefer than the first. The third, when it occurs at all, may last only a few minutes before the brain drifts back toward lighter NREM or into the early preparations for NREM. The delta waves in these later episodes are lower in amplitude, not absent, but diminished. The discharge of sleep pressure, visible in the very architecture of the waves themselves, their height falling in proportion to how much of what was owed has already been paid. By the time the sky outside the window begins to carry the first gray suggestion of morning, the slow wave work of the night is largely done. The circuits that generated it are still present, still capable of producing the same large slow waves they produced in the first hour of sleep. They simply no longer need to. The homeostatic clock has been reset. The synaptic landscape of the cortex has been adjusted. The day's metabolic residue has been moved a little further from where it accumulated. The growth hormone pulse has done its work in the periphery, and the morning's tissues are a little more recovered than the night's initial state would have predicted. The immune cells that circulate with heightened activity in the small hours of the night have completed another cycle of their patient internal surveillance. None of this is visible in the sleeping phase. The stillness of the deep sleep hours and the lighter stillness of the early morning hours, look much the same from outside. The body does not signal its own restoration. It accomplishes it without ceremony, and the only evidence that it occurred is the state of the brain and body that wakes, more capable, more alert, more metabolically ready than they would have been without the night, and carrying no memory of the work that made them so. The slow oscillations, the long sweeping waves that defined the night's deepest hours, will return tomorrow night. They will return the night after that, and the night after that, for as long as the brain is healthy and the body sleeps. The thalamic reticular nucleus will fire again. The relay cells will fall quiet again. The cortex will generate its great slow waves from within itself, each one traveling from front to back across the sleeping brain in the same direction as every other night, at the same slow frequency, the rhythm older in its form than the individual brain that produces it, a rhythm shared by every sleeping mammal that has ever drawn the darkness in around itself and let the outside world go still. The room is quiet. The darkness holds. The breathing is slow and deep and regular, each breath arriving and departing at the pace of something that needs no encouragement to continue. The slow waves move through the cortex, one after another, in the dark, each one a little lower than the last, as the first hours of the night give way to the middle hours. The amplitude of each wave, carrying its own quiet measure of what the brain has already done and what remains, the long, slow accounting of a night's deep rest proceeding to its natural end. The thalamic gate holds